Hello, this is Dr. Morris, and I'm continuing the lecture series with Chapter 16, and this talks specifically about non-specific defenses of the host being the human body. Now, non-specific defenses, I think it's important for us to differentiate between pathogenic microorganisms and the normal flora that we see on the human body. Now, if you recall, we have normal flora, which is also referred to as normal microbiota, and then we have organisms that can cause disease. These are pathogenic microorganisms, and they're endowed with special properties that, given the right opportunity, it enables them to cause disease. Now, our body has a number of defenses against these microorganisms, and in general, our ability to ward off disease through our defenses is called resistance. And then our vulnerability, or lack of resistance, is known as susceptibility. Non-specific resistance refers to the defenses that tend to protect us from any kind of pathogen. And then we have specific resistance, or an immunity based upon antibody production. This is a defense against a particular microorganism. Now, I would like to also differentiate between antibody production, antibodies that are produced inside the body by our immune system, and then antibiotics, which are synthetically produced. These agents or these drugs are administered in response to a bacterial infection. Typically, antibiotics that are made synthetically only are made to treat bacteria. They are not made to treat things like viruses and fungi, which may enter our body. Antibodies that are produced by the immune system, however, form against antigens. Any type of foreign invader, whether that's a viral or whether that's a fungal element, whatever the foreign invader is, our antibodies produced by our immune system can fight those off. Now, there are also some mechanical factors in the host defense. The intact skin, which consists of the dermis, an inner, thicker portion composed of connective tissue, and the dermis, an outer, thinner portion consisting of several layers of epithelial cells arranged in continuous sheets. The top layer of the epidermal cells contain a protein, keratin. Intact, these barriers are seldom penetrated by microorganisms. Now, there's also mucous membranes. Mucous membranes line body cavities that open to the exterior. These include the digestive, respiratory, urinary, and reproductive tracts, all of which are openings to the outside environment, all of which have mucous membranes. Mucous membranes consist of an epithelial layer and an underlying connective tissue layer. Cells in a mucous membrane secrete mucus, which prevents these cavities from drying out. If the cavities were allowed to dry out, then these cavities would crack and also would allow for infectious organisms to enter into the system. Now, another mechanical factor involved in the protection of the lacrimal apparatus is the ability to manufacture tears. This apparatus has a cleansing effect on the surface of the eye. Many of the products, I might say, that are produced or washed away by tears may be viral agents, they may be bacterial agents, but the tears that are produced by the lacrimal apparatus also produced a antibacterial substance that sometimes they can be bactericidal. That's just an aside that I figured I'd mention. Moving to saliva now. Saliva is produced by the salivary glands. This washes microorganisms away that enter the respiratory and digestive tract. Cells of the mucous membrane of the lower respiratory tract also contain structures referred to as cilia. You may remember cilia from our discussions in anatomy and physiology, and we also discussed cilia earlier in the lecture series for microbiology. These are microscopic hair-like projections that move in a synchronous manner. They propel inhaled microorganisms that are trapped in the mucus out of the respiratory system. This is also referred to as the ciliary escalator, in which the cilia, in this wave-like synchronous motion, passes mucus out of the respiratory center, where it either is expectorated in sputum or it can be swallowed into the digestive tract, where the digestive enzymes in the stomach will destroy these microorganisms. Now, the flow of urine also provides another form of a defense for the host. The flow of urine tends to remove microorganisms from the urinary system. You will find when we discuss the urinary system specifically that it is the length of the urethra that also can provide a part of a host defense. The male urethra, for example, is longer than the female urethra. Thus, males tend to get less urinary tract infections than females. If you remember, we talked about bacterial growth cycle of microorganisms. Bacteria tends to grow and multiply, and as it grows and multiplies in the urinary system, in the urethra, it slowly advances toward the urinary bladder. In the male, bacteria that enters into the external urinary meatus has to go through several growth cycles before it would be able to reach the urinary bladder. Because the length of the male urethra is longer than in the female, it would take bacteria a much longer period of time to multiply and make it into the bladder. The male would have multiple opportunities to urinate, and thus the stream of urine would wash the bacteria out of the urethra. In the female urethra, it's shorter. It's only 2 to 3 centimeters in length. And because of that, one growth cycle, perhaps two, the bacteria has already grown and migrated into the urinary bladder. Thus, females have a greater incidence of urinary tract infections than males. Now, there are also vaginal secretions that tend to move microorganisms out of the female body. This can add as another host defense. The vagina also contains a number of normal microbiota, a part of the normal vaginal flora, which also compete with other invading organisms which would tend to cause an infection or disease. Because the normal flora inside the vaginal mucosa competes for the same nutrient source, many bacteria and fungi, such as Candida, don't grow because the normal flora tends to compete for the same nutrient source. Another aspect of nonspecific host defenses is the epiglottis. The epiglottis covers the larynx during swallowing. Nonspecific host defenses, just as a review. Remember, we talked about the term susceptibility. This is a lack of resistance to a disease. Resistance, the ability to ward off disease. Nonspecific resistance, defenses against any pathogen. Specific resistance is immunity, resistance to a specific pathogen. Also, just as a review, as we talk about non-specific resistance, remember, first line of defense, intact skin, the mucous membranes that line the openings into various systems, such as the oral mucosa, vaginal mucosa, rectal mucosa, the normal microbiota, as I discussed in the urinary, in the vagina. This normal microbiota would compete for the same energy source. We see normal microbiota in the oral cavity, which also competes against organisms that would tend to cause the disease, but because they have the same nutrient source, are established normal flora, the bacteria that normally exist in our mouth or in any of these areas, competes for the same nutrient source, and thus would prevent opportunistic infections that would cause opportunistic organisms which would cause infection to be able to be impeded. Now, for the second line of defense, we have to remember to look at things such as the phagocytic white blood cells. Now, remember, white blood cells are a part of the inflammatory response we see in this system. The phagocytic white cells are the ones that get in there, grab the antigen, and destroy it. In the inflammatory response, we see blood vessels that dilate. That's to allow white blood cells and other inflammatory cells to come into the area to be able to provide additional antibodies in that area to fight off infection. The fever response is a part of a secondary line of defense. Increases heart rate. The fever increases heart rate and allows, again, rapid circulation into the area. And then there are a number of different antimicrobial substances that are inside the system that will also provide both a systemic response and a localized response as well. And then the third line of defense, again, involves the immune system, where we see specialized lymphocytes, B cells, T cells, and antibodies specific to the infectious organism to be able to fight off infection. Again, mechanical factors would be the skin, the epidermis, and then the protective layer of the skin, the keratin layers, which is a very protective protein as well. Mucous membranes, the ciliary escalator, the lacrimal apparatus, the saliva that's produced, urinary flow, flushing out the bacterial organisms, then vaginal secretions. Vaginal secretions can be acidic, which sometimes will fight off certain that will fight off certain microbials as well that are not resistant to the acidity of the vaginal secretion. So another mechanical factor that adds to the non-specific host defenses. Now, some chemical factors. Sebaceous oil glands of the skin produce sebum. This is a form of a protective film that's over the skin surface, composed of unsaturated fatty acids. Sebum inhibits the growth of certain pathogens. Also, the low pH, as I talked about with the vaginal secretions, a low pH of the skin, partly due to the secretion, the sebum that's produced. Body odor is a result of decomposition both of sloughed off skin and sebum secretion, especially by commensal bacteria. In some commensalism, one organism benefits while another one is unaffected. Sweat glands produce perspiration. Perspiration flushes microorganisms from the skin surface. Perspiration also contains lysozyme, which is an enzyme that breaks down cell walls of gram-positive bacteria. The enzyme is also found in saliva, tears, nasal secretion, and tissue fluids. Gastric juice, mixture of hydrochloric acid, enzymes in the mucus with an acidity of a pH of about 1.2 to 3. These stomach secretions destroy many ingested microorganisms and toxins. Blood contains iron-binding proteins or transferrins that inhibit bacterial growth by reducing available iron. Other chemical factors, fungostatic fatty acid, the sebum as we talked about, low pH, again pH 3 to 5 on the skin, the lysozymes in perspiration, the low pH 1.2 to 3 in gastric juices, transferrin in blood to find iron, and then we also find certain agents can inhibit ATP production, such as nitric oxide. We also have, as a part of the host defense, microbial antagonism or competitive exclusion. I mentioned this a little bit earlier when I talked about the normal microbiota or the normal flora. These bacteria that are a part of our system that don't cause infection, don't cause disease, they live in a harmony with our human body. These organisms will also compete with organisms that may cause infection, and thus they compete for the same energy source, and by doing so it enables these microorganisms that typically would cause infection can't grow in our system because of our normal microbiota. Normal microbiota or competitive exclusion can often prevent colonization of pathogenic organisms. Blood fluids called plasma, the cells, the cell fragments of the blood are formed elements. Most important for this discussion are the leukocytes. Leukocytes are the white blood cells. Decreased leukocyte counts is called the leukopenia, and so individuals with leukopenia may have difficulty fighting off certain infections. Differential white blood cell count detects leukocyte numbers and changes, and we do that in laboratory studies. We look at the number of cells. There are a number of cells which are felt to be a part of this white blood cell response. We'll talk about some of those cells in a little detail later on. Now, here are the other cells. The three categories of cells that are part of that white blood cell response are leukocytes. Leukocytes are the white blood cells. Three categories of cells: granulocytes, lymphocytes, and monocytes. Granulocytes have granules in the cytoplasm, are differentiated into neutrophils, which stain pale lilac color with a mixture of acidic and basic dyes. Basophils, which stain typically blue-purple with the basic methylene blue. Eosinophils, which stain red. That says red, but it should be red. They stain red or orange with acidic dye eosin. Neutrophils, also known as polymorph leukocytes, also referred to as polymorphs or PMNs. All of these cells are part of the inflammatory response. The agranulocytes have granules which are not visible by light microscopy. Neutrophils, which account for about 60 to 7% of the leukocytes, are important in phagocytosis. That's important to know. Neutrophils being important in phagocytosis. Basophils, 0.5 to 1% of leukocytes. They release histamines. Remember, we talked about histamines in the inflammatory response. This is a factor that in inflammation and in the allergic response. And eosinophils perform some phagocytosis, but it mainly attaches to the outer surfaces of helminth or parasites and discharges a lethal peroxide. Now, there's a couple of test questions in this, one of which is the eosinophil response. Again, we see a great eosinophil response in helminth parasitic infections. That's an important factor to note. So if you see a person and in their differential in the white blood cell count, you see a high number of eosinophils, then you know it's a helminth or parasitic infection. Also recognize that basophils, which only account for about 0.5 to 1% of leukocytes, are important because they release the histamines. And again, histamines are important factors that you see in the allergic response. That's important to know. And then neutrophils, which account for about 60 to 70% of leukocytes, important in phagocytosis. So 3 key points, 3 key functions of the 3 cells, the leukocyte cells. These are important when we talk about the differential. Again, eosinophils increase the number during parasitic infections, and you also see an increase during allergic response as well. Lymphocytes are important to specific immunity. They are not phagocytic. Remember that. They are lymphoid tissue, or they occur in the lymphoid tissue. Monocytes also lack granules. They are phagocytic only by maturing into macrophages. This is just an illustration of the different types of cells that you can see in the inflammatory response. The granulocytes, stained, polymorphs, the other term referred to this, basophils, and then eosinophils. Percentage that we see in each of the cells, again, neutrophils predominate. Again, just as a review, neutrophils phagocytic, basophils produce histamine, eosinophils toxic to parasites. And I should bring up as well the lymphocytes are involved in specific immunity. So for those cells, you should be familiar with neutrophils, basophils, eosinophils, and lymphocytes are the important ones. Now, granulocytes and monocytes migrate to sites of infection. Granulocytes are mostly neutrophils that wander in the blood. They can pass through capillary walls and they can reach trauma sites. Macrophages are highly phagocytic cells. They're called wandering macrophages because of their ability to migrate. Fixed macrophages are histiocytes. They enter tissue and organs and remain there. Examples of these are Kupffer cells. These are located in the liver. Fixed macrophages, referred to as mononuclear phagocytes, are a part of the reticuloendothelial system. Granulocytes predominate early in infection, as indicated by the differential count. As the infectious subsides or infection subsides, then we'll see monocytes will predominate. Mechanisms of phagocytosis: chemotaxis and adherence. Adherence are the attachment between the cell membrane and the phagocyte and the organism is facilitated by what's referred to as chemotaxis, which is the attraction of microorganisms to chemicals. Opsonization: this is the coating of a microorganism with plasma proteins such as antibodies and complements which will promote phagocytosis. Now, we also see in this mechanism of phagocytosis is the ingestion and digestion process. Following adherence, projections of cell membranes of phagocytes or the pseudopods engulf the microorganism and then fold inward, forming a sac around it. This is called the phagosome or the phagocytic vesicle. The process is called ingestion. This is within the cell. Enzymes containing phagosomes and lysosomes of a cell will fuse and form larger structures called phagolysosomes, with which you see the bacteria is usually killed pretty quickly. Enzymes in the lysosome include lysozymes. There's various hydrolytic enzymes. They're called myeloperoxidases. Their action or process is called an oxidative burst. Indigestible material in the phagolysosome is called residual body, and it moves to the cell boundary where it's discharged. Some microbes evade adherence by phagocytes with structures such as M proteins or capsules. Others may be ingested, but they're not killed. This is the example produced substance called leukocidins and other complexes that actually kill the phagocytes. Still others can survive inside the phagocytes and continue to multiply. The word phago refers to as eat, cyte refers to as cell. Ingested microbes are particles by a cell performed by phagocytes. Now, operating within the body, the host's second defense, which are the phagocytosis, is an important element. Again, the word phagocytosis derived from the Greek word eat and cell, which refers to ingestion of microorganisms or any particulate matter by a cell. Phagocytes are blood cells or derivatives of blood cells that perform this function. In the process of phagocytosis, this illustration is taken out of your book. Again, you see a process of chemotaxis or adherence of microbes to the phagocyte. We see the ingestion of the microbe. You see a process by which the phagosome is formed. Fusion of the phagosome, which has lysosomes, form a phagolysosome. You see digestion of ingested microbe by enzymes. You then see the formation of what's referred to as a residual body containing undigestible material, and then you see the process by which the discharge of waste material, a process of exocytosis, occurs. Remember, early on in step 1 is referred to as endocytosis, the bringing in of the organism, and then the release by exocytosis. You should be familiar with these steps involved in phagocytosis. Different types of organisms and their processes. Again, the M protein, as we talked about earlier, which inhibits adherence. We see organisms Streptococcus pyogenes and Streptococcus pneumonia has M proteins. We see other organisms have leukocidins. Staphylococcus aureus has leukocidins. This basically kills the phagocytes. Thus, the organism would be resistant to phagocytosis. We also see certain organisms which will lyse the phagocytes as the membrane attacks the complex. Listeria misogynes is an organism that can do that. Some can escape the phagozymes or phagosomes. Shigella is an organism that does that. And then certain substances prevent phagosomes and lysosomal fusion, such as in the HIV virus. And then in the case of Coxella burnetii, we see it survives the phagolysosomal process. Now, another part of the host response is inflammation. Inflammation is a host response to tissue damage characterized by redness, pain, heat, swelling, and perhaps loss of function. In general, it's beneficial because it serves to destroy the injurious agent and confines or walls it off to allow repair or replacement of damaged tissue. So the inflammatory response, the inflammatory host response, is very important, and again, it can be beneficial. Now, as we think about inflammation, we think about the redness, the heat, the pain that it generates, and we may think that's a negative impact, but it does serve a very important purpose in the body. Some of the processes that we see in inflammation, vasodilatation being one of them. We see this in the first stage of inflammation. It involves an increase in blood vessel diameter. Therefore, more blood flow gets to the injured area. We also see an increase in permeability, which allows the defense substances in the blood to pass through the walls of the blood vessel. Vasodilatation is also responsible for redness, heat, edema or swelling, pain, and inflammation. Substances that are released at the site include histamines. This is released by injury. increases the permeability as well. Kinins, which cause vasodilatation, are also increased permeability and attract phagocytes. Again, in the inflammatory response, you should be familiar with these key findings: redness, pain, heat, swelling, and or edema. In the acute phase, proteins are activated, include the complement, the cytokine, and the kinins. Vasodilatation, a response to histamines, kinins, prostaglandins, and leukotrienes. The process of margination, which we'll talk about more in just a minute, and the immigration of white blood cell counts to the point of injury, and then tissue repair. Again, these chemicals released by damaged cells in the response. Histamines lead to vasodilatation. Kinins also lead to vasodilatation. Prostaglandins, the intensity of histamines and a kinin effect. Then leukotrienes, which increase the permeability of blood vessels as the phagocytes will attach. Other substances that we see released include prostaglandins. These substances are released by damaged cells, and leukotrienes, which are substances produced by the cells related to prostaglandins. They cause increased permeability of blood vessels. They help to attach pathogens to phagocytes. Clotting elements in the blood help to prevent the spread of infection. A localized collection of pus, which is basically dead phagocytic cells and bodily fluids, in a cavity is referred to as an abscess. Now, within an hour of beginning inflammation, phagocytes will appear at the site. Blood flow decreases as the phagocytes stick to the inner lining of blood vessels. This process is referred to as margination. Then it passes through the vessel wall to the damaged area. This immigration is referred to as diapedesis. So again, two processes need to be familiar with these terms. The first of which is margination. Again, blood flow as the phagocytes stick to the inner lining of blood vessels. This is referred to as margination. And then these phagocytes will then pass through the blood vessel wall to the damaged area. The immigration of these white blood cell counts is referred to, or white blood cells is referred to as diapedesis. So you need to be familiar with these terms. Monocytes appear later in the inflammatory response. They mature into macrophages later. Macrophages ingest dead tissue and invaded microorganisms. So you need to know the function of macrophages, again, to ingest dead tissue and invading microorganisms. The final stage of inflammation is tissue repair. This involves the production of new cells by the stroma, which is the supporting connective tissue, and the parenchyma, which is a functioning part of tissue. Scar tissue results in stromal type of repair. Now, in the fever response, the hypothalamus controls body temperature. Normally that's at 98 degrees Fahrenheit. Setting can be altered by the ingestion of gram-negative bacteria. Sorry about the typo there. Gram-negative bacteria, resulting in the release of endotoxin, which causes the release of cytokines called interleukin-1. Another fever-inducing cytokine is called alpha tumor necrosis factor. It's produced by macrophages and mast cells. Cytokines are immune system chemical messengers that will be discussed later on. A chill, which is cold skin or shivering, is a sign of fever and rising temperature. Crisis refers to the very rapid fall in the temperature. Fever are often beneficial and aids the body tissue repair and inhibitors of microbial growth. This is the inflammatory response, and again, you should be familiar with these components. Again, you see damage. Bacteria enters into the tissue, gets beyond the initial protective layer of the skin. As this bacteria, the example is used, a knife penetrates the skin, gets into an area, basically injects bacteria under the epidermis, and bacteria will start to proliferate. The tissue response to tissue damage: chemicals such as histamines, kinins, prostaglandins, leukotrienes, represented by the blue dots we see here on the picture, basically released into the damaged cells. We see blood clots will form to stop off any bleeding that occurs in this incident. then an abscess starts to form under the skin. You see the process of margination which occurs as the white blood cells will line along the walls of blood vessels, and again these are fenestrated blood vessels, so then you'll see the white blood cell counts through a process of immigration. Diapedesis will occur, and we'll see these white blood cells move to an area where they can attack the invaded bacterium. Again, the final part of that process after phagocytic migration and phagocytosis is eventually tissue repair. Again, this is just being a little redundant. Hypothalamus normally set at about 37 degrees Celsius, which is 98 degrees, 98.6 degrees Fahrenheit. Gram-negative organisms produce endotoxins that cause the phagocytes to release another chemical substance, interleukin-1. The hypothalamus releases prostaglandins that reset the hypothalamus to a high temperature. Body increases its rate of metabolism by shivering and raising the temperature. When we see interleukin-1 is eliminated, the body temperature will fall. That's referred to as crisis. A part of the immune system that's important to understand is referred to as the complement system. The complement system consists of a group of about 30 different proteins that are found in the blood. Now, the blood serum is a liquid portion of the blood that remains after it's drawn, and the clotting proteins form a clot, which are formed elements. Complements participate in the lysis of foreign cells. Okay? And it's very important that we have this process take place. Again, complements participate in the lysis of foreign cells, inflammation, and in phagocytosis. The system can be activated by the immune reaction. There's a classical pathway, an alternative pathway, and then a newly discovered lectin pathway. Is the classical pathway and the alternative pathway, which we'll focus more on. Complement proteins act in an ordered sequence. This is referred to as a cascade. One protein basically activates the other. The protein C3 plays a role in both the classical and the alternative pathway. In the classical pathway, activity is initiated when the antibody molecule binds to the antigen or the bacterial cell, and they use that bacterial cell as an example. In the alternative pathway, does not involve antibodies. Complement proteins, the proteins are called factors B, D, and P. P standing for properdin, combine with certain microbial polysaccharides. Now remember, the polysaccharide coating is what we see coating the bacteria, especially affected lipopolysaccharide cell wall portions, the endotoxins such as of gram-negative enteric bacteria. In the lectin pathway, we see macrophages stimulate the liver to release certain substances such as lectins. This enhances optimization in the binding of the cell and carbohydrates. Cytolysis. In this process, complement proteins then bind to two adjacent antibodies and initiate a sequence known as a membrane attack complex. Circular lesions called transmembrane channels. Membrane pores are formed and cause eventually the lysis of a cell to which antibodies are attached. In the process of cytolysis, remember, complement proteins then bind to two adjacent antibodies and initiate a sequence known as the membrane attack complex. So it's important that you recognize and remember that definition. Inflammation also develops from the complement system, or the complement activation. Other complement proteins combine with mast cells and trigger the release of histamines, which increases the blood vessel permeability. One protein chemotactically attracts phagocytes to the site. Optimization, again, if those of you who don't know that definition yet, need to commit that to memory. Opsonization or immune adherence. It promotes the attachment of the phagocyte to the microbe. Complement involvement results in interaction between special receptors on the phagocytes. Interferons are another chemical substance that's produced in the body. Interferons are of three types. Alpha, beta, which are produced by virus-infected host cells, and gamma, which is produced by lymphocytes and kills bacteria and protein. Remember, alpha and beta produced by virus-infected host cells, and then gamma, which is produced by lymphocytes and kills bacteria. Now, they tend to interfere with viral multiplication by inducing the uninfected cell to manufacture messenger RNA for the synthesis of antiviral proteins. Now, one example is synthase, oleoadenylate synthetase. This degrades viral messenger RNA, which is another protein kinase which inhibits protein synthesis. So commit that to memory as well. You need to know the three types of interferon: alpha and beta produced by virus-infected host cells, and then gamma, which is produced by lymphocytes and kills bacteria. Interferons are important in the protection against acute virus-caused infections. The example given is influenza. Interferons can be produced by biotechnology, a method that's been tested to determine if antiviral and anti-cancer effects. The three types have different effects. Again, alpha interferon, beta interferon causes the cell to produce the antiviral proteins and inhibit viral replication. Gamma interferon causes neutrophils and macrophages to phagocytize bacteria. So alpha beta virus for inhibiting the virus, and then the gamma interferon particularly for bacteria. This illustration is out of your book, and it basically just shows the steps by which interferon works. Again, the virus typically replicates into a new virus. We see the viral RNA from the infected virus cell enters the cell, uses our cell's own DNA to carry out this replication. The infected virus also induces the host to produce interferon, again alpha and beta interferon. And then we see interferons released by the virus-infected host cell, which will bind to the plasma membrane or the nuclear membrane receptors of the uninfected neighbor host cell and will induce them to synthesize antiviral proteins, or AVPs. This includes the enzyme I discussed earlier, oleoadenylate synthetase, and protein kinases. New viruses are then released by the cell-infected neighboring host cells. We see the antiviral proteins then will degrade the viral messenger RNA. This will inhibit protein synthesis and thus will interfere with viral replication. And this is a key step. Again, if we can degrade the viral messenger RNA, remember, we need the messenger RNA, or the viral element needs the messenger RNA to be able to carry out protein synthesis. Without protein synthesis, you get no replication.